Good afternoon, everyone, and welcome to our coffee seminar today. My name is Anna Coltino, and I'm a Professorial Research Fellow in Crop Sciences here at QAFI. And I have the pleasure of facilitating this session, which is on International Women's Day. So let us start with the acknowledgement of country. I acknowledge the traditional owners and their custodianships of the lands on which we meet today. I pay respect to their ancestors and their descendants who continue cultural and spiritual connections to the country. We recognise their valuable contributions to Australia and global society. Okay, we have a couple of housekeeping things for today's seminar. You know it's scheduled from 12 to 1. And after the presentation is concluded, we'll hold a question and answer session. If you find you have questions that you'd like answered as you listen to Emma's talk, please use the Q&A button and please do not use the chat button, and we will address your questions at the end of the seminar. So now it's my great pleasure to introduce to you Dr. Emma Mace. She is an associate professor at the University of Queensland, and she's also a principal research scientist at Queensland's Department of Agriculture and Fisheries. I've known Emma for over 10 years, and I have the pleasure of working with her on a Bill and Melinda Gates funded project called High Gain. Emma's research interests lie in the development and application of innovation genomic tools to support plant breeding programs. She has a particular focus on the grain crop sorghum, and Emma's been involved in a range of projects to increase the rate of genetic gain in sorghum in improvement programs both in Australia and internationally through work, for example, in Ethiopia, which has been funded by the Gates Foundation. Emma's other passion is mentoring women scientists, and she has been involved in a range of activities to promote women in science, including the Australia Awards Women's Leadership Initiative. So without further ado, I'd like to introduce Emma now. Could you please start your seminar? Thank you.
Well, thank you, Anna, for your lovely introduction. And I'd like to start by saying I too acknowledge the traditional owners and their custodianship of the lands on which we meet today and pay my respect to their ancestors and their descendants. I'd like to thank the QAFI seminar series organisers for giving me the opportunity to present today, particularly on this day. In acknowledgement of International Women's Day, I'd like to start off with some brief thoughts and reflections on how the status of female leadership in science has changed over the course of my career, before moving on to the main talk content, which focuses on one element of my research career, which has been to develop and deploy innovative genomics resources to support plant breeding. So I started off by looking into the history of International Women's Day and was very interested to find out that International Women's Day originated more than 100 years ago, with the first International Women's Day gathering in 1911, supported by over 1 million people in Europe. In Australia, International Women's Day has been informally celebrated since 1920, with the Whitlam government officially recognising the day in 1974, in line with the UN starting to celebrate it in the International Women's Year of 1975. IWD is a combined celebration with Mother's Day in a number of countries, and in other countries, they mark Mother's Day in different times in March. And finally, each year, International Women's Day festivities have an official theme. This started in 1996, where the theme was celebrating the past and planning for the future. This year's theme is Break the Bias. So I thought I would very briefly consider one view of the bias through the lens of my 30-year career in science. I finished my first degree at the University of Nottingham in the UK in 1993, went on to do a master's and a PhD at the University of Birmingham, and then 3 postdocs. The first at ILRI in Kenya, one at UQ with Ian Godwin, and lastly one at ICRISAT in India. before returning to Australia almost 20 years ago to take up a research scientist position with the Department of Agriculture and Fisheries, formerly DPI. And then 5 years ago, I also took up a joint role with UQ. And now my time is divided as 80% UQ and 20% DAF. So for ease of a comparable benchmark, I focused on the 3 universities that I've been part of over the course of my career. And one of the metrics I was keen to look at was how female leadership in science has changed over the last 3 decades in these different universities. So I started off with various web searches, but quickly realized that only very recent data, for example, just over the last few years, was readily available. So I made contact with HR departments and other contacts I remembered from my time at these universities. So looking at the University of Nottingham, I had some excellent correspondence with the current and former Deputy Pro-Vice Chancellors for Equality, Diversity and Inclusion. And between us, we could recall that the number of female professors across a range of science departments back in the early 1990s was not significantly different from 0. The HR department confirmed they only had data going back to 2015. This raises an issue also relevant to my professional discipline of plant breeding, that if you don't measure it, you can't change it. Regarding the University of Birmingham, I had more luck going through former tutors Brian Ford Lloyd and David Marshall, and also the former head of school from my time at Birmingham, who managed to dust off the electronic cobwebs from his old data archives and pull out some data for me on the number of permanent female academics in the School of Biological Sciences in the 1990s. So not professors specifically, just female academic staff numbers. I've converted these to percentages of the total academic staff in that department here. And you can see that by the end of the 1990s, the numbers were climbing to around 10%. But once again, the HR department was not able to provide any data going further back. Records just don't appear to exist going this far back. I'm delighted to say, however, that UQ was a refreshing change. And I was able to fairly easily, with the help of Todd Sullivan and Nicole Barton, get access to 2 decades worth of data on percentages of females in specific positions across all departments and institutes at the university. I focused on levels D and E and compared the percentage university-wide wide levels with the percentages in science-focused departments and institutes. So looking first at level D, which is the associate professor level, in this graph here, the purple line is the UQ-wide line and the red one is the science-specific. And you can see that there's a pretty good increase over the last 2 decades. There's a similar story with level E, that's the professorial level, but the percentages are smaller. This graph plots the percentages for science for level D and E on the same graph to more clearly show those differences. So today we're sitting at around 20% of science professors at UQ being female and 30% of level D scientists being female. So how does this compare with current figures worldwide? I found a nice report published on International Women's Day last year on women in higher education, published by UNESCO, which had produced this rather lovely graphic looking at a few key metrics summarized globally. I realize it's too small to see the details, so I've just pulled out a few for us to look at. Starting with comparing figures for females and males achieving bachelor's and master's level education. And here you can see that women, which are represented by the bars on the right-hand side of the cap, actually make up a slightly larger share than males. However, this drops down at the PhD level and drops even more if we look at PhDs in science, where only around 25% of science PhDs are female. 2 more comparisons I wanted to highlight. The first is the percentage of females and males in research positions in universities. Overall, 30% of the world's researchers are females. So here UQ is achieving well above this level, with having 30% of associate professors scientists being female. The last metric is that of publications, and again shows quite a skew towards males versus females. I also found this reflected in a recent study looking at a historical comparison of gender inequality in science, which again showed the pretty large difference in percentages of authors that are male versus female. It was an interesting paper, and they broke this down further into countries, with Australia certainly not the worst offender, but still not at parity. Interestingly, Australia does not rank particularly well in the Global Gender Gap Report published last year, ranking 50 out of 156 countries overall, but ranking top for educational attainment. Now, by focusing on the percentage of females in high-level positions in universities, I fully appreciate I am looking at one slice at the extreme end of a complex pipeline. And there are many points along what is termed the leaky pipeline that result in women not progressing in their career in science. I will end this brief preamble to my talk by acknowledging that UQ has put in a lot of strategies and policies to tackle the lack of gender parity in research, including the Athena Swan programme and the Mole Pledge, which has been taken by a lot of UQ staff to request gender equity as a condition of participation in any panel or conference. Additionally, collecting and analysing data on gender inclusion is a critical first step to making change. As scientists, this is our bread and butter. And as Lord Kelvin, an eminent 19th century scientist, said, to measure is to know. If you cannot measure it, you cannot improve it. So I'm going to use this as my segue into the remainder of my talk, where I'll talk about one element of my research career, which is to develop and deploy innovative genomics resources to support plant breeding. The crop I have worked the most on over the last 2 decades is sorghum, which is a tropically adapted C4 grass species, the world's 5th most important cereal crop, a close relative of sugarcane, maize, and millet. It has a pretty small genome and high levels of genetic diversity for many traits, and is adapted to a wide variety of environments. I created a bit of a tongue-in-cheek title for my talk today, comparing the process of developing new elite sorghum lines with beneficial sets of alleles and genes to panning for genetic gold. Another phrase people often use in this context is finding the needle in a genetic haystack. But what plant breeders need to do is search not through one haystack, but through a whole farmyard full of haystacks in order to identify relevant genes from a broad range of material, including wild relatives, and stack them in suitable combinations in elite lines. With recent technology developments over the last decade, we now have a range of new omics approaches we can use to help us with this panning for gold, to find valuable genes from a range of different material. So first of all, let's look at what's the challenge? Why do we need these new technologies? How do we use them? One of the major issues is that increasing the rate of genetic gain for complex quantitative traits such as yield, drought, heat tolerance remains a major challenge for crop improvement programs. Why? Well, in order to breed for them, we need to understand the genetic factors, their interactions with the environment that underlie such complex phenotypes, and to use this information to increase the response to selection. This response to selection can be influenced by 4 main elements as defined in the breeder's equation. And genomics can impact on all elements of the breeder's equation by helping to select for increased genetic diversity and to increase useful phenotypic variance, by increasing selection intensity, by increasing the accuracy of the heritability measure, and by decreasing the time required from making a cross until using an individual from that cross as a parent. However, of course, Genomics alone will not maximize the response to selection. Instead, what is required is a set of interlinked and complementary resources that can be used in multiple ways to support activities in breeding programs. Today, I'm going to talk you through a set of interlinked resources that the Sorghum program based at Hermitage Research Station in Warwick has developed, which includes a range of genetic populations, data resources, software tools for use in crop improvement activities, which when used together, offer opportunities to rapidly increase the response to selection. What I'd like to do now is focus on individual elements one at a time. In the majority of cases, the work for developing these resources went across multiple different projects, so I won't highlight all the relevant projects specifically, but I will point out when work was related to a specific project in particular. So I will start by going through the genetic populations. To make use of the genetic diversity in the sorghum gene pool, we have developed a range of genetic resources that span the genetic variability within the sorghum gene pool from the wild and weedy germplasm through to highly adapted elite germplasm. This includes a large diversity panel consisting of over 1,000 individuals from diverse geographies, covering all the major racial groups of sorghum, color-coded in the PCA as shown here, with the racial groups forming the peaks of the spinning pyramid. And this resource is an excellent point of entry to explore native variation in the species. We have also developed a large nested association mapping population, consisting of both wild and diverse exotic parental lines crossed to an elite adapted line, with over 50 populations totaling 5,000 individuals. The parental lines are highlighted in this second PCA, and you can see that there is less diversity captured than in the diversity panel, but it provides an unusual opportunity to look at these exotic alleles in the context of an elite genetic background. Finally, we also have multiple elite populations from pre-breeding trials at advanced yield testing stages, color-coded here as blue for the males and pink for the females, to give you an idea of the more limited diversity of these elite lines in comparison to the other genetic populations. But what is particularly powerful is the interlinked nature of these three populations and how their complementary diversity levels, resolution, that is how near we can get to the underlying gene, their power and their suitability for performance testing in farmers' fields. So the diversity panel provides high resolution, but only low power, and is not so suitable for performance testing in farmers' fields due to a larger variability in height and maturity, whereas the NAM and the pre-breeding lines provide us with high power and suitability for testing in farmers' fields, but the NAM provides slightly more resolution than the pre-breeding lines. So depending on the situation and the trait in question, we can use these different populations in complementary ways to help us answer specific research questions. Let's now look at the data resources element, which, when combined with those population resources we've just looked at, allows us to identify genes underlying complex phenotypes through a P to G or phenotype to genotype approach, as well as understand how the functional gene changes impact on the phenotype through a genotype to phenotype approach. Let's have a look at some of these data in more detail, starting with some of the sequencing resources and first the pan-genome. What is a pan-genome? So the pan-genome represents the entire set of genes within a species, consisting of a core genome containing sequences shared between all individual of the species and the dispensable genome. In other words, a pan-genome refers to the universe of genome sequences existing in a species. It was first conceived almost 2 decades ago for bacteria, but was applied to plant species only 8 years ago. And since then, a pretty large number of plant pan-genomes have been developed. To develop a pan-genome, significant investment has to be made in developing high-quality genomes of multiple individuals. So we need to be clear about the benefit. With the rapid evolution of hardware and chemistries that have increased throughput and dramatically brought down sequencing costs over the last decade, it has recently become feasible to generate high-quality genomes of multiple individuals within a species. And by doing this, it is becoming increasingly clear that the use of a single reference genome per species can be very limited, and that pan-genome analysis offers opportunities to overcome limitations of using a single reference genome through the investigation of the entire gene repertoire present within a particular taxonomic clade. The figure here is adapted from one of our recent reviews on the utility of pan-genomics for crop improvement. On the left, we have a single reference genome shining a light on a limited area of the total gene pool of a crop species. That is, we're only able to see what is present in the reference genome. On the right, by replacing a single reference genome with multiple reference genomes, we are able to capture substantially more of the variation in the entire gene pool. The number of individual genomes needed to capture the entire repertoire of genes in the gene pool is dependent on a range of factors, including the biology and the evolution of the taxonomic lineage, and they can be categorized as either open or closed pangenomes. So what can a pangenome tell us that a single genome can't? It can help us identify multiple types of functional gene content variation, including presence-absence variation, where a gene or genes is present in one individual but absent in another, copy number variation, which can identify different duplication levels across genomes, as well as other types of variation which impact on gene functionality through both SNPs and indels, insertions and deletions, in key features of the gene. Our sorghum pangenome was based on 16 carefully selected diverse lines designed to capture the majority of the likely variation within the primary gene pool of cultivated sorghum. I'd like to acknowledge that this work was led by Yongfu Tao, a fantastic postdoctoral scientist in our group, and we've recently published this study in Nature Plants. So the spinning PCA plot highlights the selected lines color-coded according to the racial groups and adaptation to different geographies within Africa, set within the context of the diversity panel I showed earlier. This set of 16 also includes interfertile wild relatives from Africa. We sequenced using a combination of Illumina and PacBio libraries, resulting in around 150 gigabases of data or 200x coverage per genome. I will say that in the 6 years since we started this project, the technology and platforms have changed vastly. And if we were to start again, we would do so with a very different set of platforms. It's a very fast-moving area. So one of the key interests with this data was to look at gene variation within the cultivated gene pool. We identified just over 60,000 gene families in total, with gene number per individual genome varying from around 32,000 up to 37,000. So a fair amount of structural or presence-absence variation was clearly identified. We then wanted to look at how these gene families were distributed across the genomes and found that around 1/3 were identified as core genes. That is, they were present in the vast majority, and here we define that as at least 14 of the 16 genomes. A further 1/3 was defined as shell genes and identified between 2 to 14 genomes, and the remaining 1/3 was identified only in one genome and defined as a cloud gene. We can then explore the differences between these three classes of genes, and we found that they have different structural elements between the groups, with core genes being longer and with more exons than the other two types of genes, and with shell genes found to be more variable and having higher non-synonymous synonymous substitution ratios, or KAKS, than core genes, supporting their faster evolution and that they are less functionally conserved. We were also able to explore the differences in gene function between these gene classes through comparing frequencies of GO or gene ontology term enrichment, and found that dispensable genes were significantly enriched with those terms involved in biological processes such as defense response, secondary metabolic processes, in comparison to core genes, which were enriched for core biological processes, including reproduction, system development, and seed development. Overall, we were able to build up a more in-depth picture of the genome landscape of sorghum that enabled us to identify features of the genome with higher proportions of genes affected by structural variation, and in particular found genes in recombination elevated euchromatin regions, that's the green element in lane A of this figure, were less likely to be affected by large effect mutations compared to heterochromatin regions. And that we could identify higher levels of genetic diversity in these euchromatin regions in comparison to heterochromatic regions, which gives us insights into driving forces creating genetic variation and purging deleterious mutations. So in summary, the development of the pan-genome within sorghum is allowing us to address a range of research questions, including insights into the genome dynamics between wild and cultivated sorghums, in addition to insights into structural variation in gene networks. We're now working on a new project in the ARC Centre of Excellence for Plant Success to extend our sorghum pan-genome and generate an anthropogeny or grass pan-genome, including not only additional sorghum de novo genomes, but also genomes from grass relatives. I've highlighted the additional species we'll target, that is maize, which diverged from sorghum around 9 million years ago, and rice, which diverged from sorghum around 50 million years ago. This will be the first cross-genera pan-genome to be developed and will provide a new comparative genomics resource to help us identify shared and unique genes among and within these three important cereal crops and investigate evolutionary history of gene networks in the grass family. We're working with a team at QFAB at UQ to provide bioinformatics support, to develop the pipelines to predict the genes across the genomes, and look at functional enrichment and diversity across the gene families. The pan-genome projects that I just described all rely on de novo sequence generation, which is still pretty expensive. A related but cheaper technology is called resequencing. And this allows us to look at whole genome sequence variation, but mainly just SNP variation in a much larger number of lines. Almost 10 years ago, a group of researchers in both DAF and UQ, including Ian Godwin and David Jordan, together with collaborators at BGI, developed the largest sorghum resequencing resource globally at that time, looking at 47 sorghum genotypes representative of all of the racial groups. And we generated high confidence data on over 10 million sequence polymorphisms, which we can use to look at the patterns of diversity across all chromosomes and at all genes in the genome, including chromosome by chromosome sequence diversity, the number of SNPs per chromosome. And over the last 10 years or so, we've been mining these data at various scales to allow us to look at variation genome-wide at any gene in the genome of interest. In the last 10 years, the cost of resequencing have also dropped dramatically. And recently we have increased our resequencing resource 20-fold and have resequenced our entire diversity panel with my colleagues David Jordan, Yongfu Tao, and Adrian Hathorn. We're now mining this resource across a range of projects looking at both fundamental and applied research questions. Another data resource I'd like to show you is the Sorghum QTL Atlas. Again, this was a world-first QTL resource that we developed for the global community. But first, let's step back and explain why there was a need for this type of resource. Over the last 2 decades, hundreds of QTL and GWAS studies have been published in sorghum, producing an enormous amount of information concerning the genetic basis of quantitative traits, including the genomic location, allele effects, interactions with other genomic regions. But making use of this information is not straightforward, because there is no central repository of QTL studies that allowed users to compare results of all current studies against a common framework. And the absence of such a centralized repository was likely due to the complex nature of the data and the heterogeneous nature of QTL studies themselves, leading to variable reliability of QTL identified from one study to the next, meaning that significant time investment would be required to integrate the information and generate a comprehensive and high quality QTL database. In 2009, David Jordan and I published the first study that attempted to not only consolidate, but also adjust QTL confidence intervals, that is their size, based on parameters in the original study. At that point, the database consisted of just 44 studies, 700 QTLs and 160 traits. We aligned them all to the first version of the sorghum genome and to the sorghum consensus genetic linkage map, which I developed and published with David Jordan also in 2009. It was a good start, but of course, by the time it was published, it was already out of date. So together with the help of my wonderful colleague, Tracy Shatty, we've continued to update this resource. And 10 years on, in 2019, we published an updated study and created an accessible database online consisting of almost 200 studies, 7,000 QTLs for over 200 traits. And we've aligned these to all of the new versions of the sorghum genome that has been published since 2009, as well as whole genomes of maize and rice to facilitate comparative genomic studies. This resource we've developed has enabled researchers to answer a lot of questions which would have been very difficult previously, including what other QTL are nearby my trait of interest that could be affected by any selection I apply? And in what populations of QTL for my trait of interest been identified? I'd like to highlight one example now focused on comparative genomics and the utility of investigating the syntenic locations in maize and rice by making use of the known synteny across species and to link in with the work we're doing on the grass pan-genome. So this slide shows the 10 chromosomes of sorghum going around in a clockwise direction. The next 3 lanes, B, C, and D, show the comparable or syntenous regions from rice and maize. For any given QTL location in sorghum, we can predict the syntenic location in maize and rice. And linking to whole genome assembly resources for both species, we can identify all of the predicted gene models within the QTL-CI in both maize and rice. As an example, I've highlighted the location of the gene Phi-B within a QTL for maturity on sorghum's chromosome 1 and the predicted syntenic locations in rice and maize, both of which also contain the Phi-B gene. And the atlas links to external resources for these species that enable the user to further interrogate these genes. Now, in the atlas, we also make use of previously published resources that provide information on which genes within the QTL-CI are under selection, not only in sorghum, taking information from our 2013 study, but also in comparable studies in rice and maize. And one of the main reasons that we've included this information is because it allows us to take advantage of known gene order conservation or synteny between closely related species. And the fact that there are 4 times as many QTL and GWAS studies in rice and maize compared to sorghum. And by doing this, we can make use of information gained in other species, compare locations across species, and compare genes underlying QTL across species. We're now working with a group from Cold Spring Harbor who are developing a new centralized hub for the sorghum genomics community, Sorghum Base, to make our database accessible through this site. Moving on to the 3 types of data detailed here, that is genotypic, pedigree, and phenotypic, the main point I wanted to make was about the growth in the volume of these data types in the last 10 years by our program. In particular for genotypic data, points giving us increasingly higher coverage of the whole genome, and also the phenotypic data, which is increasing exponentially due to the development and use of new sensors and platforms in our group and with our expert pilots and data processors, Ken and Sean, shown here. So these data are an essential part of our approach to better understand and enhance complex quantitative traits through both a phenotype to genotype and a genotype to phenotype approach, with the volume of data generated being high. And because of the high value of these data, we need to manage it well. All of the genotypic, phenotypic, and environmental data generated by our activities are stored in linked or linkable databases. This has a number of advantages, including reduced cost of research, increased pace of research, and reduced time to impact for industry. What I'd like to do now is briefly focus on analysis and use of the data to address both a range of fundamental and applied research questions. And I'd like to start off with this overall schematic about how we approach the identification and prioritization of potentially useful traits to select for inbreeding, which starts with an understanding of the production environment and the identification of important complex trait targets. And based on our knowledge of trait biology and through trait dissection and simulation modeling, we can then identify candidate traits that can be used to screen a number of our populations to search for genetic variation for these traits. When favorable variation is identified, we can feed this information back into the crop model to predict the consequences of this variation in different genotype by management scenarios. I thought a real-world example might be helpful here, and I'm going to focus on root angle. So Graham Hammer's group, together with David Jordan, a number of years ago, conducted studies on nodal root angle in sorghum and observed that changes in the angle are likely to influence water capture in different environments by modifying both the horizontal and vertical exploration of the soil, and that modeling with best bet assumptions indicated that substantial yield gains in some environments were possible if the correct root architecture was deployed in a specific management by environment context. So knowing the potential importance of the trait, we first needed to know what variation existed in our germplasm. So a screening method was developed by Vijaya Singh in her PhD and used to screen a number of diverse lines, which included parent lines of a number of our interlinked populations. And having found significant variation, a more high throughput screening platform was developed and used to screen multiple populations. This work was conducted across 2 specific projects. The first, a joint BMGF and ACR project led by David Jordan and Graham Hammer, and the second, a joint ACR and IDRC project led by Andrew Borrell. We are now in the process of finalizing the across population and across experiment statistical analysis being led by Colleen Hunt. And once we finalize the analysis from the more recent experiments conducted in the IDRC project, we will be able to combine it with the results from the first project, where we identified QTL, integrated it with our QTL atlas and our resequencing resources, and have a look at these elements. But the more interesting thing that I wanted to highlight was how, with the integrated nature of our resources, we can access pre-existing phenotypic data, including yield and staging across these screened populations, and hence explore the association between nodal root angle and yield in more detail. We are also able to mine existing performance data on breeding populations across environments to better understand the trait value and the impact on yield, and to combine with simulation modeling to identify which alleles contribute to better yield performance across environments. Really what this rather complicated figure is showing us is a set of interlinked resources, which when used together, allow us to conduct a much more powerful study and have more immediate impact than the use of any one piece separately. So this example shows how we have used genome resources as a linking technology to enable us to answer a number of important questions, such as, are the genes for root architecture segregating in my populations? Am I selecting for a particular root architecture? And what impact does a particular gene for root architecture have in a specific environment? This particular example has also used simulation modeling as a linking technology to consider a number of what if questions, including what root architecture QTL alleles would work best in my current environments and management systems? And what is the likely variation or risk associated with growing the best combinations? I'd like to give one more trait-specific example for the application element of my talk. This one focused on grain size. This work arose from an ARC discovery project together with Ian Godwin, David Jordan, and Jimmy Vitella. To give you a brief background on the trait, grain size is a key yield component of cereal crops and a major quality attribute, both for grain growers in terms of percentage of small grains and for grain users in terms of impact on processing. Processors want bigger grain, but also consistent grain size. Growers ideally want bigger grain and increased yield, but at the very least, bigger grain without yield being compromised. So why would yield be compromised? Well, there is a trade-off between grain number and grain size, where as grain size increases, the grain number decreases. But variation in grain weight, independent of number, still exists. But this trade-off is complex, and to access this slice of the variability requires understanding of the different factors contributing to grain size. So grain size in cereals is a function of both the potential maximum grain size as well as the capacity of the plant to fill the grain. While the potential maximum grain size is largely genetically predetermined, the capacity to fill the grain is strongly affected by assimilate availability. So we developed a phenotyping treatment to try to separate the capacity to fill the grain from the potential grain size by removing half of one of a pair of selected heads within a plot to remove assimilate availability as a constraint. Yongfu applied this phenotyping treatment to both the diversity panel and the NAM resource that I described earlier, and then developed a processing pipeline to measure a number of grain attributes. Oops, sorry, I need to have gone forward too quickly. Okay. Yeah, so a processing pipeline to measure a number of grain attributes on thousands of lines. He then mapped the grain size measurements across the two treatments and across both populations and identified 50 QTLs across both populations and treatments, with HH denoting the QTL identified in the half head treatment and FH the QTL in the full head treatment. By comparing across treatments, 21 QTLs were detected only in half heads, which are more likely to be associated with inherent genetic potential of grain size that is affected by source availability, whereas 18 QTLs were detected only in full heads and are more likely to be associated with the capacity to fill the grain. With the identified QTL, we could then integrate with our other genomics resources to identify candidate genes. I've shown one of the candidate genes here, GS3, which is a well-known gene controlling grain size in rice and which is highly expressed in sorghum in early fluorescence, and which we found to be under selection during sorghum domestication. GS3 increases grain size, but appears to have little or no impact on grain number. By identifying putative causal variants in the gene through mining our resequencing resource, we can use these as targets for gene editing or gene transformation approaches. And the function of GS3 is currently being validated by Ginny Patella's group. But we're also working on a number of other gene editing targets with Ian Godwin and Karen Massel. Finally, but very briefly, I also wanted to highlight two other ways genomics tools can be applied in breeding programs. And that is via a traditional marker-assisted selection process for a specific trait or a specific region of the genome, which, when dealing with a large effect SNP, such as disease resistance, can be an effective approach, all through genomic selection, using all data points from all points in the genome at one time and predicting the phenotypic response based on statistical models. And our group has done quite a bit of work on investigating prediction algorithms for sorghum across genetic backgrounds and environments. So to summarize, I've given a brief overview of the resources and tools we have developed, with a major focus on the genomics resources and how we use them in a range of research projects, with two trait-specific examples of how we use them. I wanted to highlight, however, that there are a range of traits we are either working on currently or have in the past using these approaches. And to finish off, I wanted again to stress that the real power comes when you employ an integrated approach to build bridges across scales and disciplines. And to use this approach to understand context dependencies, to operate in relevant context. We focus on exploring natural genetic diversity and at ensuring we work at sufficient scale for the problem, and to maximize interdisciplinary research and data sharing between groups. The work I've described is multidisciplinary, and I'm so very fortunate to work with an amazing team. And here I would like to acknowledge and thank the team in Queensland that spans both DAF and UQ, with genetics work led by David Jordan, who has an amazing vision to integrate islands of knowledge. It's a privilege to work with this team. I would also like to acknowledge and thank funders through the Queensland Government, through DAF, through ACR, ARC, the Crop Diversity Trust, the Bill and Melinda Gates Foundation, IDRC and GRDC. But as this is International Women's Day, I would like to finish on this slide and to acknowledge, thank and celebrate the amazing women I work with and who enrich my life both professionally and personally. With that, I'd like to thank you for listening, and I will hand back to Anna to manage the questions.
Thank you, Emma, for a fabulous talk. I mean, we could dissect lots of elements of that all the way through. That's the resources that has been generated for sorghum is fabulous. If I can start with a series of questions, let me see if I can group some of these. Okay, you mentioned that if you were to start again on sequencing the sorghum genome to use a different set of technologies, could you elaborate on that a little bit, please?
Sure. Well, it is a very fast-moving field, and the changes that have happened, particularly with a new technology called Oxford nanopore technology, mean that you can get very long reads and very accurate reads now. So, as well as new technologies and platforms such as Oxford nanopore coming, there's new chemistries with PacBio. It's also a lot cheaper. So, if we had the same set of resources, the same budget, we would be able to think about doing things in a very different way. We could potentially do more samples, or we could get greater coverage, or we could do further follow-ups with other elements to really increase the quality. It's unfortunately, it means that by the time you publish something, you're almost guaranteed that what you're publishing is a few years out of date.
Sure. You also mentioned, I think, what approach or tool are you using to develop the grass pan-genome? I think you mentioned that for the ARC.
Yeah.
Can you comment briefly on that too?
Yeah. So we are using a range of really high quality de novo genomes that have been published for rice and maize, obviously, as well as our own sorghum de novos, as well as generating more sorghum de novos as well. And we're working with QFAB to develop a pipeline where we take the assembled genomes and develop a standardized gene prediction approach so that we can then have a standardized platform that we'll use for the cross genomics comparisons. We don't want to just take the assemblies as they are and the gene predictions. We want to ensure consistency, and hence we will need to take a few steps back, but we will be using existing de novo sequences generated from other groups.
Okay, thank you. There's a comment here that it's interesting that the original sorghum reference genome, BTX623, has the lowest gene number. And is there any evidence that gene number variation is predominantly in large gene families?
Well, that is a good question. And I think that our gene family analysis that we've been able to do with the pan-genome allows us to look at that in a very different way than if you just had BTX623. The number of genes also, by the way, identified in BTX623 has changed quite a bit from version 1 of the genome that was published in 2009 to version 3, which I think was last updated about 7 years ago. So I think that there is certainly, we have the resources in place to look at that question a lot more now.
Okay, thank you. Another question, grasses contain a large number of LTRs, on-terminal repeat sequences and retrotransposons, which are sources for novel genetic variation and I guess gene expression. Is there much variation in content of these genetic elements between various sorghum accessions?
Yeah, Sorry,
secondly,
yeah.
are they likely to be influencing gene expression and perhaps giving, novel protein coding genes or functions? It's a fairly big question.
It is a big question, and it's a fairly short answer because we haven't had the opportunity yet to look at that in detail. So predicting these, but we will be doing that in future because we're also combining with some methylomics work that we'll be doing with Peter Chris that will allow us to look at unmethylated regions of the genome and build up a more complex picture and to identify these, the regulatory regions in the genome to look at these. But right now, I don't have an answer, but it is something that we have on our list of things.
Thank you. More towards a sorghum question on perennial grain sorghum. The question is, how can pan-genomics and other omics be deployed to regenerate economically viable perennial grain sorghum? And do you know who is working on perennial grain sorghum in Australia? Yeah.
So there are obviously a number of companies that are developing perennial sorghum. As a research question, this isn't one that has been prioritised by research funders for investigating, but we have done some research, and also I have collated quite a lot of information on research that's been done internationally in the QTL Atlas. So we have knowledge of regions of the genome that are associated with rhizomes and other elements of perenniality that we could then explore. And in terms of the pan-genome, what we would be able to do there is have a look how gene content variation happens in those particular QTL regions and identify whether or not there are some types, and we do have some forage types that have a particular gene presence that are absent in the cultivated grain types. So I'd say that there's a lot of scope and we have the things in place to be able to address that.
Thank you. Now, there's one more question because it's International Women's Day that has been asked, and I'd like to finish off with that. And then we're nearly running out of time, but I think we'll be able to wrap up. So, reflecting on your own career, what are some of the persisting barriers to progressing to leadership positions for women that you can see? If you can give a fairly succinct answer.
Yes, well, that's a big question and probably the hardest one to give a succinct answer to. I mean, the obvious thing is the work-life balance. It is the obvious element that still falls predominantly on the female shoulders compared to males. And that might impact on just that slower development at the start of the pipeline that makes it a bit harder then for competing on the same terms with your male colleagues. I think that there's been an element of that in my life. I feel very fortunate that I have been working with such an amazing group of people, both females and males, who have been very supportive. But I think it depends on the context of the group that you're with and your ability to be able to really cut through and push forward. And it is possible, and I think I am also guilty of it, of not leaning into things as much as maybe some of my male colleagues do. I remember Ian Godwin telling me about this lean-in concept and how when faced with going for promotion, a woman would look at the promotion document and say, oh my goodness, I only meet 8 of the 10 criteria, I can't go for it. Whereas a man might look at it and say, oh great, I get 3 of the 10 criteria, I'm going for it. So it's about recognizing, holding back and pushing yourself forward anyway.
Okay, so I think we have to wrap it up there. There are a number of other questions which I might pass on to you after this session, I think, which might be helpful and you can answer them directly. I think we have to move to, so thank you again very much, Emma. We need to advertise, I think, the next science seminar. That is on Tuesday, the 15th of March, and it's being given by Melissa Wooderson. And it's entitled The Effect of Analgesia and Hemostasis on Beef Calves During Dehorning in Northern Australia. So please attend that. And then finally, I think there's some, the next slide, I think. Emma, oops. All right, so thanks for attending our science seminar. Here we go. Thank you for attending our science seminar today. If you haven't signed up to our seminar list, please do so at Coffee Science Seminars. And if you want to give us any feedback, please use the QR code to give us feedback. Please scan it. So this has been a wonderful presentation, and thank you, Emma. Thanks, people, for the questions. And we have to say bye-bye, I think. Thanks again. Goodbye, and thank you very much for attending the seminar.